Hey everyone. It's great to be here. Thanks for this opportunity. My name is Arjun, and my group is right now at Michigan State, and we are actually moving to the University of Colorado Archer's Medical Campus to join the Biomedical Informatics Department this August. And what I'm going so I want to first start off with acknowledging the terrific group of people that I get to work with every single day. And today, what I'm going to do is to talk about a work done by Matthew Ortruso, a terrific undergraduate student in my group. And then Matthew and I we essentially been talking about this problem of making sense of gene level results based on literature. So what I mean by that is the following, right? So the scenario that I'm going to talk about is so commonplace. A researcher essentially uses things like association studies, omics technologies, or high throughput screens, and at the end of the process, they essentially generate either a large gene list, which is a ranked gene list based on some score from the experiment, or maybe the output is simply a gene set, which is an unordered set of small number of genes. Either way, the next step that the researcher takes is to go to the literature and try and understand what these top candidates are. Any of these high-throughput experiments are totally, supposed to be, general hypothesis generating experiments. So, which means that you want to go to the research, literature and find out what is special about these top genes or the small number of genes set. And this is currently done typically through manual search and inspection right now by most researchers. And ultimately, they want essentially the researcher is trying to figure out are there biomedical terms essentially, are there tissues, phenotypes, diseases, drugs, symptoms, or whatever it is that keeps showing up for the diseases at the top of their result. Okay. So what we are interested in trying to do is to try and automate this process of trying to remove the manual inspection of literature and try to automate this identification of relevant biomedical terms as much as possible and give those results to the researcher and have them triage the results in a much more efficient way. So just to again make the problem clear, we are interested in this very general schema. The input is either a ranking of genes or a small unordered gene set. We want to plop in some automated algorithm in the middle, and then the output should be relevant terms, very broadly defined in biomedicine. Right. And ultimately, what we want to do is to literally, for example, get ranked, prioritized lists of maybe tissues, cell types, traits, diseases, symptoms, drugs, and so forth. Okay. The way we sort of, started formalizing this problem is to say that we're going to think about the biomedical data, rather the data that is hidden in literature in the form of a matrix like this, where every single row is a gene, every single column is some general biomedical term. Okay. And then a particular value across I and j, a cell here, is simply recording the association of gene I to term j, mined from the literature through some method. Okay. So the question is, what is the best way to come up with these associations? And then what does this plus sign mean, right? What is the best operation here that combines this input gene list with this text mine association to get our prioritized terms? So there are many ways of doing this. Of course, we are not the first people to think about this. There are, for example, Lars's group has been doing a terrific work over many years, working on co-occurrence-based methods, where they essentially find out gene term associations from literature and rewarding basically terms that occur in sentences or abstracts, and then coming up with some scoring scheme that ultimately gives you a single score for association between a gene and a disease or a gene and a tissue and so on. Okay. And I think there was one talk earlier in today's session where a member of Lars's group was talking about this advanced dictionary using Tagger and then using statistics to get these tissue associations. Another idea is to take this co-occurrence idea to one step further, to instead train a shallow neural network model that takes these co-occurrence statistics and then learns a numerical embedding for every single word or phrase or term. And then you can use something like cosine similarity between a gene and a term to find out associations. For example, you can go to this resource at the bottom right of the slide and then essentially get 200 dimension word embeddings of genes and terms. And one of the problems is that there are also multi-word phrases that one is interested in. And then the way we sort of deal with this is to simply average the embeddings of the individual terms. For example, if it's carbohydrate metabolism, we would just find out the embeddings for these two and then merge them into a single term. However, both these approaches have one small problem, which is that they rely on accurately tagging the genes and terms in the first place, right? So we thought, okay, can we sort of add another complementary approach to these already pretty good methods? So we came up with a very simple method, and our idea is to simply say, okay, we're going to think about genes and terms to be very similar if they return very similar results if they are searched on PubMed. That's it. Super simple. Okay. So for instance, what I mean by a term is if the term we are interested in is a melanoma, we'll say melanoma and all of its synonyms. Just say or term melanoma or synonym one or synonym two and so on. Search PubMed and get results. You will get a list of PubMed IDs that PubMed says it's a match, right? You can do the same for a gene of interest. Then you get another list of PubMed IDs, and you simply say what is the overlap between these two sets. Then you can turn this into an enrichment score. If you are interested, I can talk to you more about what this score exactly is, right? And this is simply the association, right? Okay, so that's it. So the way we do this is for terms, we literally look through all the terms in a given ontology. The method is we have implemented this in a super general way. You can plop in any ontology you want, and you can get PubMed results for every term in the ontology based on terms, names, and synonyms. We query up to the top 100k PMIDs, which is it's up to, okay. And then we restrict the returned PubMed IDs to only papers that are also have a gene mentioned based on gene to PubMed. And then we compare these two sets to get an enrichment score. So the method is super simple. Okay. So these are the three methods: Word2Vec, the work from the Jensen lab, and then our PubMed overlap. And then so those are the three methods here. And then what about this operation here, right? So what we are trying to do is to say the gene input, like I said, we want to keep this super flexible. So which means that the gene input can come as a ranked list or an unordered small set. The term input can again come as a ranked list or an unordered set, okay, which is from the bind literature. So we essentially implemented a bunch of similarity measures that will take any type of input, either rank, rank and set, a set and set across gene and term. Okay. So these are all very standard methods. So we try all of them, and then we plug this into this framework. The way we're trying to evaluate this, so we started with trying to evaluate this using GTEx data. So GTEx data comes by genes and tissues. And then we converted this data into a score where every single row and column here, a cell indicates the specific expression of gene I in tissue J. And because we know these tissues, what we want to do is to combine this, assume that we get a single column in this particular GTEx data. And we want to figure out, can we somehow find out the tissues based on literature that corresponds to the correct tissue the sample actually, the gene actually came from, okay? So what we essentially want to do is to construct this matrix where the input tissues are along the column, text tissues are along the rows, and then every single value is supposed to be the similarity. And then, for example, this is an example of an output that we get. And what we want to do is to say, go to a single column, which is the GTEx tissue, and then ask if the tissue that corresponds to the correct input is actually ranked highly based on the text. For example, in this case, esophagus is ranked the highest. The redder it is, the better. Okay? For example, the same thing, if you look at brain GTEx input, the output from this text association analysis tells you that brain and nerve are the highest ones here. So you can sort of figure out what is the ranking of the correct term, and then you get some results. Generally aggregating based on input works really, really well. So we will see, in general, this PubMed overlap-based method works quite well compared to compared to the Jensen lab work and the Word2Vec stuff. And overall, for example, if you look at one specific idea where we use the aggregated z-score, we almost get the correct issue at the rank 1 every single time. So this is actually, the simple method is actually working out pretty well. But turns out that this method is actually not working well, or none of these methods are actually working really well for very diffuse biomedical concepts like traits, okay? Because a trait is sort of a very diffuse definition. So for example, to test this, we got data from GWAS Atlas. And again, these are genes, and every single column is a trait. And every single entry is the association of gene I to trait J. And then what we are trying to find out is that if we supply one single column here, essentially a gene list from a GWAS, can we find out terms related to that trait from the literature automatically, right? So when we evaluated this using our method, it's okay. It's working to some extent, which means that, for example, invariably the correct trait is being ranked at around rank 10, but that's pretty bad, right, in absolute terms. But this is a very, very difficult problem. So we are working on the stuff, and the way we are going about working on the stuff right now is that we are trying to come up with better scoring methods for this PMID overlap, add natural language processing and machine learning steps to this. We have some experience doing this stuff. And essentially, we're going to expand this and generalize this code base to make sure it generalizes to any ontology or dictionary, whatever this biomedical dictionary is. And our method is totally agnostic to the model organism. So we're going to, the organism. So right now we are evaluating this with human data, but we can as well do this with other model organisms. And then we're trying to build out a web server. What I really want to emphasize is that this PMID overlap is not the focus of my talk. Really, I would really like to figure out if, as a community, we can build some utility to increase the pace of interpreting gene lists or gene-based results. Right. So I really want to welcome collaboration. So if anyone's interested in NLP, machine learning, software engineering, web development, and this problem, please reach out, and I'll be happy to openly collaborate and then share code and all that stuff. And my goal is to not just work with our method, but use really all methods that are out there and incorporate them into a utility. Specifically, what we would like to do is to develop something like a MyGeneList web server, where a researcher comes in and simply pastes their ranked gene list or a set of genes, and then from any organism, and they should be on the cloud, our system should basically run and return relevant terms from various ontologies. And for every single term our MyGeneList prioritizes, it should also tell us which were the genes that drove this particular prioritization, and also which papers are most relevant for this particular tissue or disease or symptom. Okay. All right. With that, we also do some bunch of work in annotating samples that are publicly available using their metadata. So if you're interested in that, let us know. You can also check out a software that does that particular annotation. It's called Text2Onto. And with that, I want to thank my group again. They're an amazing group of people to work with. Thank the funding sources, NIH, NSF. And I also obviously want to give a shout out to all the other folks in my group who are giving talks and posters at ISMB. I'm really bummed that I can't be there in person, but sure, please go talk to folks in our team. And then there are also a bunch of open positions. So please come and join us and check us out on website and Twitter as well. Thanks a lot. I'm happy to take questions.